Okay, so our next topic, we're going to talk at greater length about glomerular filtration. So glomerular filtration is this first step in the formation of urine. Okay? It occurs at this portion of the nephron that is sometimes referred to as the renal corpuscle. And the renal corpuscle is simply the glomerulus, the capillary membrane, the glomerular capillaries, and Bowman's capsule. Okay, so together, the glomerulus and Bowman's capsule is referred to as the renal corpuscle. So glomerular filtration, one of the key things to remember about glomerular filtration is that it occurs through completely passive forces. So glomerular filtration is completely passive. It's also non-selective. Okay? So it's completely passive and completely non-selective. So plasma moves from the glomerulus and is filtered and enters Bowman's capsule. And it's filtered across something that we call the glomerular membrane. Okay? So filtration occurs across the glomerular membrane. And the glomerular membrane contains 3 layers. And we're going from the blood side to the Bowman's capsule side. Okay? So the innermost layer is the wall of the capillary. Okay, so the first layer is the wall of the capillary. Underneath the wall of the capillary, or beyond that, is a small basement membrane. And then beyond that is an odd-looking membrane, but it's something that we call the inner layer of Bowman's capsule. So if we look at this, you see here, this is the afferent arteriole, right, that brings blood into those capillary membranes, the glomerular capillaries. Blood enters the glomerular capillaries and moves across the glomerular membrane into Bowman's capsule. Okay. Now, there's an area here with a box that's sort of illustrating a cutout. And with this cutout, you see the innermost layer is the layer of endothelial cells. That's just the capillary membrane, right? The wall of the capillary. Then in white is this thing that we call the basement membrane. And then on the outside is this odd-looking layer that we call the inner layer of Bowman's capsule. And this inner layer of Bowman's capsule, it looks strange because it's composed of these individual cells that we call podocytes. Podo meaning feet, because these individual cells have these very extensive foot processes that wrap around the capillaries. Okay? So this membrane is basically the spaces in between the foot processes of these podocytes. Okay? Now, the thing that these 3 layers are doing, the glomerular membrane, which includes the capillary wall, the basement membrane, and the inner layer of Bowman's capsule, these 3 layers are designed to prevent plasma proteins and cells from getting out of the glomerular capillaries. Okay? So that's really the function of this glomerular membrane, is to prevent the escape of any kind of cells, red blood cells, white blood cells, and any plasma proteins, okay? Especially albumin, because albumin is a small plasma protein. So that's the purpose of the glomerular membrane. So the fluid that enters Bowman's capsule, sometimes it's called filtrate, is identical in composition to plasma, except there's no proteins and there's no cells. Okay. So the forces involved in glomerular filtration, these are all passive forces. Okay. We don't exert any energy in the process of glomerular filtration. So the forces involved in filtration are actually some of the same forces we studied when we looked at capillary bulk flow. okay, these Starling forces, which includes capillary blood pressure, plasma colloid osmotic or oncotic pressure, right, plasma oncotic pressure, and the hydrostatic pressure in Bowman's capsule. So remember, these pressures were we were categorizing them as either pushing or pulling pressures. So capillary blood pressure is a pressure that comes from the capillary, and it's a pushing pressure, right? So it pushes fluid out. Plasma colloid or plasma oncotic pressure is a pulling pressure, right? It's a pulling pressure that's coming from the plasma proteins, especially albumin, that can't cross the glomerular membrane. So oncotic pressure is a pulling pressure. So that pressure is a pulling pressure coming from the capillaries. And then finally, the hydrostatic pressure of Bowman's capsule is a pushing pressure coming from the fluid in Bowman's capsule. So that pushing pressure pushes fluid into the capillary. Okay? So as you can see, if we're talking about GFR here, right? If we're talking about the glomerular filtration, right? Glomerular filtration is a net movement of fluid from the capillaries into Bowman's capsule. You notice that we have 3 pressures. 2 of those pressures oppose glomerular filtration. Okay? Plasma oncotic pressure and Bowman's capsule, hydrostatic pressure. These 2 guys oppose glomerular filtration. And it's actually capillary blood pressure by itself that promotes glomerular filtration. And if we look at the numbers that are drawn here, capillary blood pressure on average is about 55 millimeters of mercury at the glomerulus. Oncotic pressure, plasma oncotic pressure is about 30. Bowman's capsule hydrostatic pressure is about 15. So if we subtract these 2 opposing pressures from capillary blood pressure, we notice that we have a net filtration pressure that is adequate enough to allow for filtration. Okay. So notice how important capillary blood pressure is for maintaining glomerular filtration. Okay. It has to be greater than the combination of both of these other opposing pressures. Okay. So there is a aspect of renal function that's actually very important clinically. It's called glomerular filtration rate, or GFR, okay, the rate of glomerular filtration. And this is very important clinically because GFR is something that is used clinically to represent the number of functional nephrons a person has. Basically, it's a measure of renal function and the health of the kidneys. So glomerular filtration rate, which is simply the volume of fluid filtered at the glomerulus per minute, okay, highly depends on the net filtration pressure, which we already said is very dependent on capillary blood pressure. It's also dependent on the surface area available for filtration, right? And surface area is really how many functioning nephrons you have. And it's also dependent on the permeability of the glomerular membrane. So a healthy glomerular membrane has a certain level of permeability. If glomerular membranes become injured and scarred, then permeability will go down. So in a healthy individual, they should have a pretty predictable surface area and pretty predictable permeability. Okay? So sometimes these two factors are collapsed into a single constant called the filtration coefficient. Right? Because in a healthy person, this filtration coefficient, or K sub F, is a constant value. So as you can see, if we assume that Kf is constant, that GFR is basically a function of net filtration pressure, which means that it's basically a function of capillary blood pressure. Now this is very important to keep in mind because GFR is so very important because it is the first step in the production of urine. So anything that causes GFR to go up or down is going to affect the rate of urine production. So since GFR is so dependent on capillary blood pressure, when we are talking about the control of GFR is also greatly influenced by capillary blood pressure. Okay. So GFR is such an important parameter, like I said, because it's the first step in urine production and it will determine the rate of urine production. We highly control GFR, and there are two major mechanisms for the control of GFR. There's what's called autoregulation and extrinsic sympathetic control. Now these two mechanisms of control, it's very important to keep in mind that they have two very different goals and priorities. Okay? So normally autoregulation is what controls GFR on a regular basis. So the vast majority of the time GFR is under autoregulation. And the goal of GFR is to maintain GFR, have a stable GFR in the face of normal fluctuations in arterial blood pressure. Okay? So our blood pressure fluctuates all the time. It goes up and down depending on our mood, depending on how stressed we are, depending on the position our body is in. Now, we don't want GFR to change every time blood pressure changes. So the goal of autoregulation is to maintain GFR despite any fluctuations in blood pressure. On the other hand, the extrinsic sympathetic control of GFR has a completely different goal. The goal is not to maintain GFR. The goal is to change GFR, okay? Increase it or decrease it in an attempt to control for blood volume and blood pressure. Okay? So if you've lost blood volume and your blood pressure drops, your goal is to drop GFR so that you don't lose any more fluid. You're going to conserve fluid so that you maintain your blood volume and can recover blood pressure. So two very different systems with very different goals. Now, autoregulation we're going to talk about first. And as the name suggests, it is the mechanism for intrinsic control. Autoregulation, auto means self. The kidneys can maintain GFR by themselves. So they self-regulate GFR. So keeping in mind that autoregulation, the goal is to maintain GFR in the face of normal blood pressure fluctuations. So, as I said, blood pressure fluctuates all the time. Our blood pressure increases and decreases for a variety of reasons. So, let's say our blood pressure increased for some reason. When our blood pressure increases, that means that glomerular capillary blood pressure increases. Okay? Now, when our glomerular capillary blood pressure increases, the net filtration pressure increases, which causes an increase in GFR. Okay? This is what autoregulation is trying to battle. Okay? We don't want our GFR going up every time we have an increase in blood pressure. So the goal for autoregulation is to try to buffer these changes in blood pressure so that glomerular capillary blood pressure doesn't go up and down every time arterial blood pressure goes up and down. Okay? And the way that this is accomplished, okay, the primary way that autoregulation does this is to modify the diameter of the afferent arteriole, okay, to compensate for a change in blood pressure. So either we vasoconstrict or vasodilate the afferent arteriole in order to compensate for a change in arterial blood pressure. So let's say that arterial blood pressure drops. Let's say the body's blood pressure goes down. Autoregulation will cause the afferent arteriole to vasodilate, okay, so that capillary blood pressure does not drop, okay, and filtration pressure won't drop so that GFR will stay the same. Okay. So autoregulation in response to a drop in arterial blood pressure will vasodilate the afferent arteriole, okay, in order to recover capillary blood pressure and GFR. And the opposite is going to happen in response to a rise in blood pressure. Okay. In response to a rise in blood pressure, the afferent arteriole is going to constrict, okay, so that we are bringing capillary blood pressure back down to normal and GFR back down to normal. Okay. So this is the mechanism for autoregulation. A drop in blood pressure causes afferent arteriolar dilation, and an increase in blood pressure brings about afferent arteriolar constriction. Okay. And this is all in order to protect capillary blood pressure from arterial blood pressure. So the mechanism for this, how autoregulation operates, is it all goes back to this special part of the nephron I referred to before called the juxtaglomerular apparatus. Okay? That area of the nephron where the distal tubule runs very close to the center of the V formed by the afferent arteriole and the efferent arteriole. So this area that we call the juxtaglomerular apparatus, okay, it operates this mechanism called the tubuloglomerular feedback system, which is autoregulation. Okay? So tubuloglomerular feedback is autoregulation, and it's controlled by the juxtaglomerular apparatus. So how does this work? Now, if you notice here in the juxtaglomerular apparatus, we have two special types of cells. We have what are called granular cells, which are found on the afferent arteriole, okay? And the cells that are pushed right up against it are macula densa cells that belong to the distal tubule. Okay? So the way this tubuloglomerular feedback system works is that the macula densa cells are constantly regulating or sensing the rate of fluid flow through the distal tubule. Okay? Because if GFR increases or decreases, the rate of fluid flow will increase or decrease through the distal tubule. Okay. Now, let's say we have an increase in arterial blood pressure. An increase in arterial blood pressure will cause more blood to enter the glomerular capillaries. Capillary blood pressure will go up, and GFR will go up, and the rate of fluid flow through the macula densa cells, through the distal tubules, will go up. Okay. The macula densa cells recognize that elevation in fluid flow, and the macula densa cells will trigger the granular cells to release a vasoconstrictor that constricts the afferent arteriole. Okay. Constriction will cause glomerular capillary blood pressure to come back down to normal. GFR will come back down to normal, and the fluid flow rate through the distal tubule will come back down to normal. Okay. Now, if the opposite happens, if arterial blood pressure decreases, okay, less blood enters this glomerular capillary blood pressure goes down, GFR goes down transiently, and the fluid flow rate through the distal tubule will go down. The macula densa cells sense that decrease in fluid rate, and it stimulates the granular cells to release a vasodilator. And that dilates the afferent arteriole, bringing more blood into the glomerular capillaries, recovering capillary blood pressure, recovering GFR, and bringing this flow rate through the distal tubule back to normal. Okay. So this process of communication between the macula densa cells, the granular cells, leading to constriction or dilation of the afferent arteriole, is this tubulo-glomerular feedback system, which is the mechanism for autoregulation. Extrinsic sympathetic control is actually much less complicated. Now remember, extrinsic sympathetic control, the goal was actually to alter GFR, increase it or decrease it in order to compensate for a change in blood volume and blood pressure. So sympathetic input overrides autoregulation. Okay. And sympathetic input comes through the innervation of smooth muscle cells in both the afferent and efferent arterioles by sympathetic nerves. Okay. So these sympathetic nerves release epinephrine and norepinephrine on the smooth muscle of the arterioles, which bind to alpha adrenergic receptors, and it's going to cause constriction. Okay. So sympathetic stimulation to the afferent and efferent arterioles causes constriction, which is going to cause a decrease in GFR. So an example of an activation of this extrinsic control is in the case of blood loss or hemorrhage. So in the case of hemorrhage, we have a decrease in blood volume, which causes a drop in blood pressure. That causes the body to activate the sympathetic nervous system. You have constriction of the renal arterioles, which decreases capillary blood pressure, decreases GFR, urine production goes down, and essentially you're now conserving plasma and conserving blood pressure, trying to prevent any further drop in blood pressure. So you can see how the goal of the sympathetic system very, very different from autoregulation.